Hola. I'm a professor at Scripps Research Institute, and my talk today is going to be about how you sense touch and other forms of pressure. So I start with giving thanks. I like to give thanks to people in my lab, and this is actually a picture just from a few months ago where we had a 25-year lab reunion. So I've been running my lab for 25 years, and these wonderful people that you see are the graduate students and postdocs who are now in my lab, but also ones that have been in the lab and graduated a long time ago. So all the work that I will talk to you about is done by these students that I'm very appreciative of. So we talk about sensory biology. This is how your peripheral nervous system senses stimuli and transmits this information to the brain. As you probably know very well, that there are at least 5 senses, and these are, for example, your sense of olfaction, smell, which is a chemical. Taste is also chemical sensing. You have photon sensing, which is your sight. Your hearing is based on mechanical sensing. And last but not least is your sense of touch, which is very unique because it's distributed all over your body and also has very diverse modalities that it senses. It could be mechanical, thermal, or chemical sensing. So your sense of touch is remarkable. You can sense indentations in your skin that are 1/500th diameter of a human hair. So at the nanometer scale, which is remarkable. But beyond this sensitivity, we rely on sense of touch for our emotional health. And there's many other senses that are related to touch. And one of them, which is my favorite, called proprioception. Many people don't know what it is. It's the sense of where your limbs are in space. This is, for example, how I can close my eyes and touch my nose. If you think about it, the only way I could do that is because I have sensors in my muscles and tendons, and from how much they're stretched, I have a clear idea of what space I occupy. But this is my favorite photo that shows how amazing the sense of touch is. And I love this photo for a few reasons. One of them, I have fond memories when President Obama was in the White House, but also because it shows this wonderful idea that sense of touch is so personal and emotional to us. So in this scene, this young man comes into the Oval Office and tells the president that his friends tell him his hair looks like the president's. And Barack Obama just bends over and says, "Touch it, dude." So we often say, seeing is believing, but President Obama knows that touching is believing as well. So in opposite side of the touch is pain. And of course, acute pain is absolutely essential for our survival. It's a danger signal telling us to stay away from something. But chronic pain is when pain persists, even the stimulus that caused it is no longer there. And that's a major medical problem, unmet medical need that many people in academia and industry are working to find novel medicines for pain. But today I want to go down to the basic science and tell you how we sense all of these stimuli. And all of this start in a group of neurons called the dorsal root ganglia. They're actually on both sides of your spinal cord. They're a group of about 10,000 neurons that send little processes, antennas, if you will, all over the body as sensors to tell you what's going on in the periphery. And this is a video of a zebrafish embryo while it's developing. Those little bulbous things you see next to the spinal cord are the DRGs, and all these processes are what we call axons of the neurons extending, going all the way to the muscle and skin to get information from the periphery. It's a beautiful system and just illustrates how you do this. So this is at the organism level. If you schematically look at one neuron, a sensory neuron, what it's doing is sensing something in the periphery and sending it to the brain, right? And at the termini for these types of neuron, it has to sense something very unusual, which is physical stimuli, pressure and temperature. And how does it do this? So the sensors in biology that do this are usually called ion channels, and they're very specialized proteins, but they're really very simple. They sit at the plasma membrane, which is the fatty acid surrounding of a cell, and they do either two things. They're either closed, they do nothing, or when activated, they open, ions flow in. It's been measured that 10 million ions can go through one ion channel per second, which is remarkable if you think about it. And this kind of causes electrical activity, and the voltage of the membrane changes, and this signal then propagates from neuron to get to your brain. So think of them as the electric switch that kind of starts the electrical signaling from the skin all the way to the brain. So what we're interested then is finding, my lab was interested in finding, how do you do this? What are the ion channels that helps you sense touch? The ones that help you sense a gentle breeze or a cactus pricking your finger. So the ion channels we're looking for are specifically activated by mechanical force. So to find these, we also wanted to explain to you why is this important. First, it's a major biological question. The fifth sense, how does it get initiated? So it's a basic biological question. But also, we think that finding these receptors will give us new ways to treat pain. But what I also want to emphasize is the third point there, which is when you do curiosity-driven science, sometimes the applications can come from directions that you didn't even anticipate. So it's just important to support basic science for this reason. So Bertrand Coste, a postdoc in my lab, came to find these mechanosensors, but it's very hard to try to find it from the neurons that I described it to you. So his strategy was very simple. First, he took simple cells in a dish, and he would push on them. And this shows a little video of it. He can record the electricity going through the plasma membrane, and with the glass probe, he simply pushes the cell. And if the cell has this pressure-activated channel, he can actually record the electricity through it. I don't want to get too much technical, but he found a cell that responds to this mechanical pushing, and using various techniques, he came out with about a list of 300 genes that could be responsible potentially, we don't know which one, for this activity. And then what he did was a technique called RNAi. You can knock down one gene at a time and see if this electrical activity in response to pushing is still there. And this is actual data I'm showing you. And the reason I'm showing you is because it shows how persistent Bertrand is. What you're looking at on what we call the x-axis is all the recordings that he did for pressure-dependent activation in these cells in response to one gene being knocked out. As you can see, there's not much difference in the first 72 data points that I'm showing. To get this kind of data, he had to spend about 3 days on each candidate. So literally, Bertrand came to lab for 11 months, pushed cells in response to knocking out genes, and saw no difference. So for a whole year, he didn't get any interesting data. But he persisted, and that red dot in the bottom was the big breakthrough. It was number 72, and when he knocked it down, these mechanically activated currents were almost completely gone. So we were very excited because we had found the gene that's required for pressure sensing. And he named this piezo-1, and there's a sister called piezo-2 from the Greek piezi, meaning pressure. That's one of the cool things about finding a gene is you get to name it, because the original name was unknown family 38 member A. Okay, what do piezos actually look like? This was another interesting surprise for us. So we can, from the sequence, have a guess of what the protein will look like. And when we looked at the sequence, we saw that it has many what we call transmembrane domains. These are parts of the protein that go in and out of the plasma membrane, this fatty layer that I talked about. The reason this was exciting to us is because most ion channels pass through the membrane 2 to 6 times. This one did it 38 times. So this is more than any other protein in your genome. These are so tiny in nanometer scale that we need electron microscopy to look at their structure. And this is us and 2 other groups, for example, did, and I'm showing you the actual structure, what the protein looks like. And these 38 transmembrane proteins, 3 of them come together to form this ion channel. What you're looking at is that the middle is where the ions would flow in, and each of these little ribbons that look like telephone wires are these transmembrane domains. What you will immediately notice is that the protein has this curved shape, which is very unusual. And Rod McKinnon, my colleague, hypothesized that, maybe this is how this protein is a pressure sensor, in the sense that it's curved, but when you push on it, it straightens and lets ions in. Data from my lab and others have shown that this is exactly what happens, and what I'm showing you is the morphing of the 2 structures in a closed and open state. So closed is curved like this, and as you'll see, when you push on it, it opens and flattens, and ions can go through. I think this is an amazing molecular machine that's evolved, very specifically evolved to sense pressure. As you can tell, I love this protein. I love the structure. And I thought, if you love something very much, you want to have a tattoo of it on yourself. And that's what I did about a year ago, and I want to share it with you if you don't mind. And then I had the idea that because the gating, I can put it in a place where I can demonstrate how it works anywhere, right? In a restaurant, at a party. And so this is the idea that the protein is closed, and then when you stretch it, it opens and ions can flow in. Oh, thank you for appreciating that. So the structure is great, but we really wanted to know what does it do? And in our lab, we study this ion channel in animal context. So both in animal models like mice, as well as from clinical data from humans, we see what happens if you don't have piezo2, for example. So first I want to come back to proprioception, this sense that I said most people don't know about it, but it's very important for coordination, for walking. So for example, on the left, you see this image of someone blindfolded, tightrope walking. Obviously, you cannot do this, especially if you're not looking, without this coordination that depends on muscle stretch sensing. Last year, I was in Davos, Switzerland, giving another talk like this, and there I asked a professional musician to come and play violin blindfolded to highlight proprioception, because that's the sense he's using to know where his fingers are, for example. And remarkably, this gentleman, Yuki Wang, played this piece blindfolded for the first time in front of the audience, and I'm going to play a small clip of it for you. Oh, the sound is blocked. It's beautiful if you actually hear it. So he played it perfectly without seeing, which many musicians do actually, don't look at their arms when they're playing it, again highlighting the importance of proprioception. So what happens if you don't have piezo2, we don't have proprioception? There are actually individuals that, if they can change the slide back into projection mode, oh, that's fine. My apologies. Okay, so if you don't have piezo2, and there are individuals identified that have a mutation in piezo2, so complete null, so piezo2 is not there, and what we found in collaboration with our colleagues at the NIH is that these individuals can sense touch and can sense proprioception. And this is going to show a video of one of these individuals, again blindfolded, and asking simply for them to walk on a straight line. So all of you here will be able to do this very easily, but as you will notice, this individual has a very hard time going straight because of lack of touch and proprioception. So I will just summarize a lot of work my lab and others have done to show that piezo2, both in animals as well as us humans, is very important and essential for sensing touch, proprioception, certain aspects of pain, such that when touch becomes painful, you experience this after injury or sunburn, but folks suffering from neuropathic pain also experience this all the time, so very important clinically. Mechanical itch. We've also extended these studies to what we call interoception, which is sensing of organs inside of you. So this includes breathing, blood pressure sensing, urination. So every time you feel like you have to go to the restroom, your bladder fills, you sense this mechanical force, and this is also piezo2 dependent. As well as digestion. Another example that is in active research right now, for example, is that if you have a very large meal, you feel very full. I don't know what it is in Spanish, but all languages describe it as a very mechanical sensation. And yet most of research on this is done at nutrition sensing level. So we're using now these proteins to study how it is that stretch sensing contributes to how much you eat, when you stop eating, etc. So these proteins have become very useful to define relevant and related senses to touch. And if I go back now to the initial slide of the 5 senses, one has to ask, how many senses do we really have? Is sensing your stomach stretch or bladder stretch a separate sense than touch, although they use the same molecular machinery? Very interesting questions. So how does this research help patients? So first of all, just finding out that the individuals without piezo2 have a mutation in their sensor of touch and proprioception already helps them, because we know that the visual system works together with the proprioception, and so we can encourage them to use their visual system to help them walk in a more coordinated way. But we also are very excited for pain, for example, to find novel small molecules, for example, that could bind and block this channel, and in some topical form may be very useful for future pain medicines. But one of the things I always like to say that, curiosity-driven research, like I told you about, is great because sometimes the applications come from directions that you never expect. And when we started working on this, for example, we never expected it to be relevant to bladder function, asthma, hypertension. And so all of this research is kind of can be expanded to many different indications. So I think one of the things I kept talking about is the importance of piezos in us, in mammals. But remarkably, this ion channel is conserved throughout evolution. This just shows an evolutionary tree showing that it is present in mammals, in flies, in worms, even in unicellular organisms, also in plants. And so many of these species, the importance of this protein has not yet been studied. But one of my postdocs, Alireza Musavi, looked at this in a plant. And at first you might think, why do plants need pressure sensing? Of course they do, in many different ways. And one of them is in the soil. When the roots penetrate the soil and they experience a harder surface, they have to decide if they go through or go around it. So that's pressure sensing. And Alireza showed that piezo is expressed in plant root tips, exactly where this pressure sensing is happening. And I'm not showing you any data, but without piezo, this ability to penetrate hard surfaces is compromised. So the lesson from here is that evolution is amazing, and once you find a way to solve a problem, like how to sense pressure, we use it to sense touch, and plants use it to sense the hardness of the surface of the soil that they have to go through. I just wanted to end the last few minutes by telling you a little bit about my career, and if I may give some advice to young people, which I see there's some here. So I'm an immigrant. I'm an Armenian origin. I grew up in Lebanon, and I went to the United States when I was 18 years old. And my lab is full of people from all over the world. So I feel like this immigration and the cultures coming together is so important for creative processes like science. I found it remarkable, and I kind of feel a little bit bad that many countries, including United States, now has an anti-immigrant mentality, which I think is not very good for society. And because being from Lebanon, being a scientist was not even an option. So I was thinking I was going to go become a medical doctor, and nothing against medical doctors, it's wonderful. But it opened my eyes to the possibility, because when I started working in a laboratory, I absolutely loved it and found my passion. And I think it happened to me by such a chance way that I just encourage everyone who's here, who's young, who's exploring what to do, is to experience a lot of things, and you might find something you really love and good at it. And so that's very important to explore. In science, what I found that the most important thing is to be curious. You might think that most scientists, their number one direct goal is to cure diseases, which is true. But I actually function from asking curiosity questions. We're all curious. Kids are very curious. How does this work? How do you sense pressure? That fascinated me, and just that kind of led to all these studies that hopefully will be very useful for humanity in the future. I also like to emphasize the role of luck. Pasteur said it best, that luck is great, but if you have a prepared mind, then you're ready to use that luck. And we use a word in the lab called informed intuition. Many people think that scientists only work by this hypothesis-driven, very methodical way. But many times we use our intuition based on being informed over the years of what works, what doesn't, what questions are important, and most importantly, what questions can be answered in the next 5 years. And so I encourage everybody to accept luck. It's okay, but be prepared for it if it comes your way. And then I think what I'm doing here, there's a lot of effort, again, within the United States to cut science funding, etc. And I just think that although I would rather just be in my lab doing experiments, the recent years we found out that it's very important for us to be out there talking to the public, explaining how science works, explaining that you have to support basic science, and all of the medicines and translational come from this kind of early investment in science. And so I wanted to emphasize that as well. So thank you so much for your attention, and it's been a pleasure being here.